So
Swayam Prabha, Digital India, Educated India.
we continue. We made a break at the end of describing the Raman systems, and we are going back to the catalyst. And before going into operando in situ studies, I want to touch base on an ultimate way of doing things. It's not only the physical probing, but the chemical probing. So the idea is that in the end, when something is happening, we are having that the molecule reacts with the surface of a catalyst, and something is happening. So we have to understand the surface of it. There are different kinds of chemical probings. Calorimetry is one of them. I'm not going to get into detail, but it is very informative. And when you have very specific systems, it can really tell you a lot of things because it's measuring the heat which is associated with the absorption of different kind of chemical processes. It is very versatile because you have different kind of calorimetry techniques that can help you measuring different kind of processes. And it brings you very convenient insight on the reaction. For acid-base catalysis, microcalorimetry, for instance, is giving very, very good, very detailed report. And even sometimes they're doing combination with, for instance, a steady state isotopic transient kinetic analysis. So there are many things that can be made with the calorimetry. Maybe it's not like the first choice technique, but it really helps you getting into many details of the system. So there are chapters in our book or in many other places that you can look at. And also, the most interesting I want to share with you today is when we are dealing with probe molecules. I believe that you are also going to tell a little bit about exactly. So I want to tell for one particular case which is more related to health, but the ultimate concept, the ultimate value of it is very important. In the end, with a probe molecule, it can chemisorb, and you can interrogate the chemisorb molecule because already the Raman, the infrared spectrum of the chemisorb molecule, like we saw the other day, can tell you where it is sitting. So it can tell you the chemical composition of the surface. It can also tell you things about the reactivity of the surface. It can tell you about the number of surface sites. It can tell you a lot of things. But once that you have a molecule chemisorb there, you can hit and see how it reacts. And the molecule will react differently depending on where it is sitting down. And then you can get an additional information which is coming from temperature program surface reaction of chemisorb molecules. That is very, very important. And there are multiple cases. Sagar will give a more general thing, but I want to touch base on one of the cases which is basically about methanol chemisorption. And also we have an important difference compared to the physical probes. The physical probes, we are getting bulk and surface information. With the chemical probe, we are getting essentially surface information. Not that it is completely decoupled from the bulk, because the bulk always have a sedging, but it's not that direct, and you have to keep on insisting. So you remember that it is not the same looking at the surface than looking at the bulk. And you know that there is life on Earth, so don't be fooled by bulk analysis. In catalysis, we are typically dealing with very small particles. We are going nano. That means that in the end, the surface becomes the key performer, and the surface to volume ratio is so high that typically you can really see what is happening at the surface without being fooled. But in the end, when you are doing a reaction, the molecule will react with the surface side. And surface side typically doesn't look exactly like this. This is just a drawing. But you, in principle, don't know how it looks like. What you know is that the molecule goes there, reacts, and comes out different. And the active site can be many things, for instance, supported vanadium oxide on given support, but the active site is defined by many things. There is actually a very interesting book that is available in Zenodo. Zenodo is a website where you can upload open access papers, and it is addressing rigor at reproducibility in thermal heterogeneous catalysis. It's a very good one. It's a think tank group that was discussing about it and made a document, which is very valuable, and that you really should have it. It's very interesting. And it also tells you about the active site per se. It can be a metal, an acid, a base, an oxide. that there will be an adsorption site, but the properties are affected by the support interface, how you modify the support, other adjacent absorption sites, porosity of the system, and all these things are what ultimately shape and define the active site. And actually, they even have this interesting flow chart about how to assess and catalytic testing from the catalyst performance, about the structure performance properties, about the intrinsic kinetic. And it is going, I mean, you just can go through it and find it is in the middle of the document. One of the pages has this table, Scheme 5.1 in particular. And this is the book. This is interesting in general to have it because it doesn't explain you the techniques, but it tells you about good practices, good things. So this is something that is very important from any point of view. So chemical probes, temperatures, program surface reaction. Why is it important? Well, it is important for very many reasons. We have a catalyst, we have a particle, and then you have to calculate, you have to investigate, you have to see what is happening with this catalyst, and at some moment you will have to normalize. You will have to say, okay, I'm getting so many kilograms of a valuable product per kilogram of catalyst, but per kilogram of catalyst depends on the particle size, because the exposed fraction will be different. Say, okay, fine, granted. Then I can tell you how many kilograms I get per square meter of this active catalyst. Yeah, but not all the surface is alike. You have surface, which is just physical surface, but with hardly any reactivity. You have another surface that has more chemical reactivity. You have different kind of sites. But in the end, the one who is doing the job is not the surface. I mean, you have a surface like this table, but only this fellow is doing the job. So it's not that you have like 3 square meters of the surface, no, it's that you have one site. So when you normalize, when you quantify, that will be important, because this is somehow like saying, how many friends I have? Oh, I have 500 kilograms of friends. Okay, that piece of information is good when you go into the elevator, says maximum weight 1200 kilograms, okay, but usually you don't count your friends by kilograms or by height. No, this is 2 meters, 1 meter, I have 30 meters of friends. No, you have 30 friends, 10 friends, 20 friends. So normalization is something that makes sense to us in a daily life. But then it goes to catalysis and we forget, yeah, per kilogram, per meter, per whatever. Okay, you can do that, but you have to do something that makes sense. 300 kilograms means that you have a one big fat friend or many skinny friends or whatever. That doesn't really tell you an answer. Now, making surface chemical probe can really help you in many of these things. For instance, when you are dealing with methanol, TPSR, you are doing chemisorption of a molecule which is methanol, can already tell you the number of surface sites. How do you make it? Okay, you take your sample, you dehydrate the sample, so that you have the surface hydroxyl groups, and different kind of sites. So the first thing that will happen, assuming a very simple surface with only hydroxyls, is that when you flow the methanol over this surface, the methanol will replace the hydroxyl, so that you get a methoxy where hydroxyl was. And this is something that will be happening, so that when you start flowing methanol through your sample, no methanol comes out. Why? Well, because the methanols, they simply bind to the surface, bind to the surface, bind to the surface, until the surface starts to be saturated and ultimately it starts to go out, and then basically you get a breakthrough and the methanol gets out of the catalyst bed, which means that the catalyst is already saturated by surface methoxy species. So that if at the beginning you have the hydroxyl groups, now you have methoxy species. And whatever happened to the hydroxyls? Well, they went out. Basically, every time a methoxy replaces a hydroxy, the hydroxy with the one hydrogen group from the methanol goes out like water. So this is a typical profile that you will look if you are measuring online with mass spectrometer. Basically, this is telling you that the surface is being replaced by methanol groups. And you can integrate how much methanol has been taken by your sample, so you know how many surface sites were chemisorpting methanol. This is already telling you the number of sites. And then, out of this number of sites, you can talk about the grams of your catalyst. Your catalyst will have a number of square meters per gram, And then you can also tell how many sites you have per gram, or also, from a surface perspective, how many sites do you have per square nanometer of the surface of this material. So you are getting already an important information. And then with this number of sites, you can also calculate the activity per site, because now you are not talking about grams or meters, but about sites. And then you have molecules, so molecule is reacting at a given rate. And assuming that all these sites are active, you can always calculate how many molecules react per one site and per 1 second. And this is the turnover frequency, molecules reacting per site and per second. And that is very important because this is telling you who is doing the job and if the people doing the job are very active or moderately active or very lazy, because you can see those things. And because Sagar is more to tell you something about catalysis, I'm going to tell you something which is more catalysis related. In the area of health, in area of toxicology, there is one kind of toxicology which is the toxicology triggered by nanoparticles. And this is a reaction trigger toxicology. Is some particles are very oxidative, they have oxidative stress, and they trigger a damage in your body, in the environment, in plants, in whatever, because of such a reaction triggered by the surface of the material. Toxicology community is made by physicians, pharmacologists, mathematicians, whatever. They're not used to catalysis. So at that moment in time, we got into this community, and to them it was very interesting to know that actually the surface is important, because they found that when they normalized per square meter rather than per gram, they were getting more relevant correlations. But when we were telling them about sites, and we were telling them about the reactivity of sites, and when we were telling them about the reactive nature of the sites, correlations start to make more sense. And that was a very important development. Because here, for instance, you have a collection of typical materials that they look in toxicity community. These are referenced from the Joint Research Center of the European Union, which is kind of a national European lab for reference. They have the different zinc oxide, ceria, nanomaterials like titania, ceria, multi-wall carbon nanotubes. You could tell the number of methanol chemisorbing per gram, like millimoles, but you know the area. And then you know how many methanol molecules chemisorb per square nanometer. So you can look at these materials as they typically have been doing per gram or per surface area. Some materials have very high area, others have very low area. But actually, materials with very high area, like these two, they don't have so many sites per square nanometer. So they have a big area, but this area is not that crowded of active sites. And other sites with low area have a lot of active sites in these few meters. So maybe you have less area, but very crowded, very active. And others have big area, but less active. And that explains that, like, they found like silica is typically a lot of area, but it's not very reactive. And they didn't know why. But then they said, okay, fine, there is a lower number of sites. But because you not only measure the number of sites, but the reactivity, you can hit. And the most simple cases that will happen is that on a redox site, this methoxy group will be oxidized to formaldehyde. By hitting online on the mass spectrometer, you will see that. If it is on an acidic site, two nearby methoxy groups will dehydrate into dimethyl ether and water. So you will get dimethyl ether as a reporter of acidic sites at the surface of your material. And also, if there are basic sites, they will absorb so strongly the methoxy group that by the time it goes out, it's already burned like carbon dioxide. And you can have combinations and bifunctional sites, basic and redox, basic and acid, and get dimethoxymethane, methyl formate, but this is more complex. And in any case, these are like the key basic composition. And the process is simple. It's a fixed bed reactor where you flow the purge, and then you flow methanol, very little methanol, 2,000 ppm of methanol in argon, flow through it, you purge again, and then you heat, and you observe the products coming out of it. And we were using that for the catalytic basis of nanoparticle toxicity, because in the end, it's a catalytic phenomenon. And we were looking at a group of materials. Some of them were very clear, some of them were not that clear, and we did it with the German Institute of Risk Assessment in Berlin, the BFR. We were looking at materials with high, moderate, and low reactivity for one reason, because the highly reactive nanomaterials, they were killing the cells. The non-reactive nanomaterials, they were not killing the cell, but medium reactive nanomaterials didn't kill the cell. That was strange, and that they didn't know. So The good idea is that you bring catalysis to another world, and then you get like a very different kind of interactions. So we have several European projects in this area: Harmless, Sunshine, NanoSolvit. Nanoinformatics is the one I was coordinating. We already finished, and we were combining in silico, in vitro, and in chemical. So chemical, laboratory things, computer things about what is happening. So in vitro is basically with cells or with animals or whatever. So we were doing in vitro studies with cell cultures. And they have different kind of reactions that they typically use, like carbonylation assay to see the reactivity, EPR about the generation of reactive oxygen species, but they have a problem. They know that the very reactive nanomaterials, when you make a cell culture 24 hours, at the end of the day, most of the cells die. For non-reactive materials, after 24 hours in this cell culture, all the cells are alive. But the problem is that materials that have a moderate oxidation capacity basically didn't kill the cells. And that was puzzling because, okay, if it has an oxidative capacity, it will ultimately kill the cell. But it didn't kill them. So the first thing we did is like you are kind of entering the twilight zone. Things is not black and white, but some sort of gray, and we have to understand the reason for that gray. And we have a hypothesis, autophagy. Who of you know what autophagy is? Mm-hmm. Yeah, essentially. It's a mechanism that the cells have for protection. Like when there is damage, they basically eat themselves somehow to regenerate this part of the system which is damaged. Like a protein is damaged, they destroy it, they eat it. So the autophagy is like eating your fingers, well, eating part of you, and compensate for the damage. So we're saying, well, maybe the moderate reactive things are not killing the cell, but they are damaging the cell, and the cell are repairing that damage, which may be. Is it so? Well, we made the methanol chemisorption and TPSR of all these materials that the European projects on toxicity are doing that. So with how many sites there are, the reactivity, and so on. And then we were ranking these materials. They typically rank the materials by area. So titania, high area, they have a 200 square meter per gram. Okay. But the number of sites, of course, is very high. This more or less goes in parallel. But when you normalize the number of sites per gram by the number of meters per gram, you see that samples with very low area, they have many sites per site, or like this one, or like that one. Sometimes materials with a very high area, they don't have so many sites per square nanometer. So the surface per se was not a critical factor. And when they are talking about the dose, which dose? They like to talk about grams, they like to talk about square meters, but shouldn't we talk about the number of sites if you want to get an insight on toxicity? So we were doing that. And also because of the TPSR, some materials were showing like more acidic profile in green, oxidative profile like in red, where you may get essentially formaldehyde, or more basic, where you get CO2. And we made very qualitative cartoons, like more basic, more acidic, or more oxidative, moderate oxidative, or crazy oxidative, like copper oxide. So we have this kind of qualitative and quantitative profiles on these materials. And we were also looking at some reactions to quantify the oxidation of some molecules which are relevant of protein and cellular damage, chemical reaction, to see how these molecules are being oxidized. So somehow we got what we were calling oxidative turnover frequency, which is the capacity to oxidize molecules which are relevant to protein damage or to lipids damage. And then we were observing that some materials were very active, and of course, it's not the same if you normalize this activity per gram than per surface or per active site, because this is only the part of the material that is actually doing the job. And then we found like copper or copper iron had a very high activity per site, the same case of cobalt, but others like titania, silica had a very little activity per site. So with the people in Berlin, we sent our postdoc, Victor, to work with Veronica and Andrea, and he was doing the impact on the homeostasis of the cell, what is happening to them. So they were doing a number of metabolic assays, membrane integrity assays, just to double check if the cells die or live. And a typical assay, you having a control which is absolutely inert to see how many cells die or do not die, and then a tritone, which is a surfactant that dissolves the membrane and kills the cell. So in this case, the reference material gives like the regular release of LDH, which is an enzyme inside the cell, lactato dehydrogenase. If the cell dies and breaks, LDH gets out of the cell and you measure the presence. Usually, the control or titania, which is an inert material, don't have an effect, and the triton that dissolves the membrane, all the LDH goes out and you measure, so you know the cell is dead. And the same thing with another equivalent assay to viability, so it's viable, and when it dies, it's not viable. So everything was consistent. But the interesting thing was when we were looking at two cell lines, epithelial, macrophage-like cells, but the system basically is when we did a lot of proteomic analysis. What was happening in these cells? We were looking at 2,000 plus proteins, and there is software for that, of course. And the interesting thing was that the reactive materials like copper oxide and zinc oxide, they saw a number of altered proteins. And the less reactive materials like titania or ceria, which are they have activity, they show no damage on the proteins. No proteins were damaged. So that was interesting because they are reactive, but they're not damaging any protein. Why is that? So that was strange because an inert material is okay. A reactive material is killing and is doing damage, okay. But moderately reactive materials, they were not cytotoxic. And we were doing a lot of assays basically to measure where the autophagy is triggered, so they can detect the generation of the autophagosome. And when you do that with all the cells, basically, a long story short is that for the reactive materials, you don't get any autophagosome triggered, but because the cells are dying, so they don't have chance to do anything. When you are exposing the cells to non-reactive or very little reactive materials, the autophagosome is not triggered, they do not die, but they don't need the autophagosome. But for the intermediate reactivity materials, ceria, titania, the cells do not die, but the autophagosome is triggered, that is basically telling you that the system is compensating a partial damage. So the answer of the system is, okay, the bad ones are killing them, no reactions. The good ones are not doing anything because basically they have no reactivity, there is no need to compensate. And those with an intermediate reactivity, they don't kill you, but they are ugly, they are nasty, they are doing something that is affecting you, but they are compensating for it. So there is a damage that is being compensated. So the cells do not die, but that doesn't mean that they do not suffer. So that was a little bit the classification of these materials. The most reactive ones, basically they damage the end with the homeostasis in the cell for these two cell lines, which is in line with a very high reactivity. The unreactive ones basically have no effect, and the cells make no things to compensate damage because there is no damage. And the intermediate ones, there is some damage which is being compensated by the autophagosome. So that was like a big classification of the reactivity of these materials, which we could quantitatively associate with the chemical reactivity of them. So you have like the very bad ones that kill you, the good ones, and the ugly ones. That's why basically when we talk about this, we were talking about the good, the bad, and the ugly. Reactive, non-reactive, and partially reactive. So the good ones are inert, the bad ones kill you, and the ugly annoy you, but you can live with that. And that was basically the idea. And another application that we are doing when we are talking about toxicity of nanoparticles is like, okay, you want to avoid toxicity of nanoparticles. You want to have the nanotechnology because we need it, but you want it safe because eventually there is a release. And we want to know nanomaterials that do the job that we want as a catalyst, as a sensor, as a whatever, but we want to make sure that if they are released to the environment, they don't affect you. But at the same time, knowing the reactivity of nanomaterials is something that you can use in your favor. Because the same way that you don't want these nanomaterials to affect you or to affect your environment, you also want these nanomaterials to kill the bad guys. And who are the bad guys? Viruses and bacteria. So the knowledge that we have on this is something that we're also applying to kill airborne pathogens. Mainly this is something that we started after COVID, because then we discovered that the indoor air quality is very important, not only the chemical, but the microbiological. And then we start working on many things about how to fight these kind of pathogens, viruses and bacteria. And the reason you know well, I mean, COVID was kind of the tip of the iceberg. We got worried, all of a sudden everybody was worried about indoor air quality because the COVID killed like 7 million people in a couple of years. But every year 3 million people is dying because of lower respiratory tract infections. It's happening every day. As you are breathing, as I am breathing and speaking, we are just releasing pathogens everywhere. And we are getting those pathogens. So we have to make sure how to handle that, because this is an exposure that is happening here, in the supermarket, in the elevator, in the bus, anywhere. So we work with molecular biologists people, virologists, to generate aerosols with viable viruses. And we were making these aerosols to see how a catalyst can inactivate the airborne pathogen, because people is doing inactivation by trapping them, and then you get a filter. Okay, I have all the viruses trapped here. What do I do with that? Because this filter is full of viable viruses, these famous EPA filters, but this is a problem. You want to inactivate them, but you don't want to accumulate. So basically we were making aerosols with different kind of viruses, like this is a bacteriophage, and also some viruses like the flu virus, and even we were dealing with rhinovirus and even with COVID virus. The first thing we did is to work on which kind of aerosol will result in a virus that remains alive for hours. So this is like 2 hours of the aerosol viability, and then for this coronavirus, it deactivates, and the bacteriophage is more stable, because bacteriophages are very stable anyhow. But these were like using suspensions, and in the end we decided, okay, let us use artificial saliva, which is more representative of the aerosols which is surrounding us. And in that case, the viability of the viruses was much better. Basically, in 2 hours, they remain active in the air, which is happening as we talk. Now, once we have that, we make sure that the filter, the catalytic filter through which we are throwing the aerosol, does not affect the aerosol integrity. And after 2 hours, we observe that the aerosol remains essentially stable. And then we were working on how to oxidize these viruses. Basically, the reason of doing that is a filter where the aerosol goes through it, and we do oxidative stress damage to inactivate the virus or to kill the bacteria. The technologies that are being used currently to kill them are summarized here, and basically this is amount of energy per unit of pathogen inactivated and the time per unit of pathogen deactivated. So the time that you need can be up to 1,000 times faster or 1,000 times slower. You want something that is fast to inactivate the pathogens, but at the same time, because you will want to have these kind of systems everywhere, you don't want a system that is using 270,000 times more energy than other systems. You want a system that is energy efficient, so down here, and time efficient down here. So basically you want this technology. Which technology? There is nothing there. Well, there is nothing there because we are working on that recently. Because other techniques, they use a lot of energy, they can be efficient, but they are energy intensive and they are slow. But you do have a possibility, which is low temperature catalysis. That will be fast, and that will be very efficient in the use of energy to do that. And again, catalysis is the promise of something better. So you are very good working in catalysis, and if you take catalysis, is ideal. So basically, it's filters that do not trap the pathogens, but they inactivate the pathogen, they kill the bacteria, inactivate the virus, and they do not trap them. So that is the way they work. They will be catalytic filters like a honeycomb shape or polymeric matrices that we put within the fan and within a moderate heating to do the job. And then you do work with these molecular biology people that make a culture to see how many cells get infected. And we can explore how efficient this is. The plaque formation unit is the way of measuring, so that you will observe that basically there is no plaque formation or there is a lot of plaque formation. So you can do that. And then what we are observing is that for the nanomaterials that we are using on either polymeric or ceramic filters, the efficiency is very good. And for different bacteria and virus, you can see the effects. So for viruses, we have rhinovirus, flu virus, we even have COVID virus. And basically, you are killing 1, 2, 3, 4 orders of magnitude down to here, from the fresh to the treatment. That is at least, because this is the detection limit. So that means that the system in general very efficient. And in the case of the bacteria, like with Escherichia coli or Staphylococcus aureus, you kill everybody. So that is very convenient, and that is based on the catalytic effect. So the bacteria get killed, you see a living bacteria and a dead bacteria which is broken, and all the inside is out, and you can really tell that there is a damage. So doing these kind of experiments with viruses of bacteria is very intense because you have to make a culture, you have to expose to the nanoparticles, and mainly when you are dealing with COVID, that requires very complex security protocols. So These experiments are not fast. They may take like 12 hours to 2 weeks. They're not economic because you need a lot of security, you have to work with dangerous coastlines, and that means biological risk. So these people at the Molecular Biology Center, they were working at Biological Safety Laboratory Level 3 because of the COVID. So this is like dancing with wolves. Basically, you don't want to dance with wolves, only the necessary amount, because these are my colleagues and I don't know who is who because it's difficult to tell. So how do you make a better catalyst to inactivate virus and bacteria? Okay, we run some experiments, we get some results, and then we can use the catalytic touch, probe reactions. There are several reactions that represent damage which is relevant to damage to proteins, to damage to lipids, and then you can make, with the carousel reactor, you can probe like 12 nanomaterials on these probe reactions and get a chemical reactive profile. For instance, DTT is dithiothreatol, which has a sulfur-sulfur bond, which is very relevant to protein damage. And what we are doing is to measure the progress of that reaction. And then we can make a correlation that we start to populate with the chemical capacity, the oxidative potential of these catalytic materials, so that it will go from 0 to 100% oxidation of this dithiothreatol, for instance, representing a protein damage, and correlate that with the inactivating power of the pathogens. This is a logarithmic scale. So you can see how materials that have a moderate oxidation of dithiothreatol, basically they kill about maybe 1 order of magnitude of the bacteria or the viruses. Not that good. But in this case, it's for a rhinovirus, for the flu virus, I mean. So they don't kill much. But when you have at least 70 or more % conversion of dithiothreatol, these materials are inactivating 1, 2, 3, almost 4 orders of magnitude of this virus. Now, that is nice because once that you start to populate this cloud, you can make a lot of chemical probe analysis with many formulations. And those that have a high oxidation capacity for dithiothreatol, then you take them for the biological assay, and they say, okay, these are already good candidates because we have now a chemical screening and a correlation. And from time to time also to take someone that doesn't behave so well to verify that indeed it's not so good at inactivating. And then you can populate something, and out of simple chemical analysis that you can make in 15 minutes with 12 samples, you can already pre-screen and pre-select reactive materials which are more relevant to apply on the inactivation of viruses and pathogens. Okay, so we are working with a chamber like this, but now we are working in a chamber much bigger, the size of basically an elevator, where we can have like bigger aerosols and use more representative things and put our catalytic there with more. So this kind of work is not easy. You need all these virologist people. He was this winter in the South Pole looking, this winter and the previous winter. He has been a couple of winters looking at avian flu in penguins, and maybe you saw the news. He was working on reporting a lot of avian flu in the South Pole, and he was the person doing that. And we are working with him and his people on this kind of virology. Our people in Madrid and also another group in our institute doing the same thing. We're doing more thermal, and these people is doing more photo, because this is something very ambitious, and that is the idea. Surface chemistry, are you talking on rotary work on iron molybdate, or can I tell it? Or are you having that in your slides? Okay. No, iron molybdate is a very nice system because in the end you're interested on catalysis, on chemical engineering, but air-door quality is something that will be very important because we're talking about chemical air quality that I didn't talk yet, and microbiological air quality, but in the end, the technology has to be catalytic. Filters trapping viruses and bacteria is a headache because you have viable ones, and then you have to get rid. And this technique is better because a catalytic filter never saturates, keeps on running forever. Virtually. If we go back to catalysis, iron molybdate is a very interesting catalyst used, for instance, for the oxidation of methanol to formaldehyde, and it is a bulk catalyst, essentially. But like every bulk system, it has a surface, and when you look at the surface in detail, the details of the surface tell you something. You remember that I was telling you that the surface of a material is like a catastrophe, that you have a periodic thing that regularly repeats ad infinitum, and all of a sudden what happened? Well, what happened? It's the end of the world, it's the end of my universe, there's air outside, and what happens? So the surface changes, and you see that it looks different. You see like a regular pattern, and things don't look that regular, they're like some kind of amorphous thing, and this is like the outermost nanometer or 2 nanometers, so something is happening there. And actually, we were commenting yesterday about the low energy ion scattering, that you can measure what is happening at the surface, and with several scans, the iron molybdate in the first scans, you see that there is a lot of molybdenum that is more or less leveling off, but iron was not visible, and scan after scan after scan after scan, iron becomes more and more visible. Actually, when you plot the ratio of iron to molybdenum against the number of scans, you see that the presence of iron is increasingly visible. So it means that even when this is iron molybdate, the surface is essentially molybdenum. And when you are still knocking off, say, it is iron molybdate. But at the beginning, from the outside, it looks like molybdenum only. And actually, even when you make element profiles, you can see that there is maximum of concentration of molybdenum at the edges of the particles compared to the middle. And of course, along as you go there, you see that the iron has minima at the edges because you have a surface enrichment of molybdenum. So the surface, what the molecules see, where the reaction happens, is mainly molybdenum. Again, surface and bulk, so keep that in mind. If you look at the catalyst at the different references samples, it's very easy to tell with Raman. Molybdenum will look like that. You have other bands, but 820 is a very characteristic one. Iron molybdate has this one at 780. It's very clear. And then when you look at the catalyst with different amounts, you see the two peaks. Maybe more molybdate than molybdenum, or similar molybdenum than molybdate. That depends on the position, but basically this is molybdenum and this is iron molybdate. So you can make a map. You can look at the surface of these catalysts, which are pellets. They have this form like a ring, like that, a raspberry ring. And then you can cut it and look what is the profile of these materials. And then what you observe is that on the fresh catalyst, you have a distribution where most of it is basically more the Raman band of iron molybdate, and then you got some molybdenum here, but then after the use, you get a much more even distribution of molybdenum and iron with a higher presence of molybdenum all over the system. So the higher exposure of molybdenum in this kind of catalyst is something important. And actually, when you look at the reactivity, methanol oxidation is an oxidation reaction. Methanol TPSR on iron oxide is essentially producing dimethyl ether. It's an acidic material. Of course, methanol dissolves and reacted, it always happens, but as a reaction product, you get DME, dimethyl ether. And you are using this iron molybdate catalyst to make formaldehyde. So the iron per se does not appear to be a key performer. And you already seen, according to the low energy ion scattering, that after several scans, and here you have 300 scans, the first sample was essentially moly, but in reality it is iron molybdate. But this is not what the molecule sees. What the molecule sees is essentially molybdenum and the surface. So you have a catalyst which you mix iron and molybdenum to get iron molybdate, the green one. But at the end of the day, iron per se, you don't have it segregated. You will have iron molybdate always. But the surface, the outermost layer, no matter how the particles are, will be essentially molybdenum. So why do I need the iron for? Why don't I use simply molybdenum to do this reaction? Because in the end, if you look at molybdenum TPSR, you will get, of course, a little bit of unreacted methanol that comes out, but essentially formaldehyde. And actually, this is a catalyst for methanol to formaldehyde. So molybdenum is all you need to run this reaction. Well, here you have even a TPSR profile of methanol on iron molybdate and on molybdenum coating iron. Well, essentially the same. Even you get formaldehyde even at lower temperature, in the case of molybdenum at the surface of iron, that in the case of iron molybdate. So it looks very much alike. Why do I need iron molybdate? Well, we're also talking about lattice oxygen mobility. And if you make several chemisorptions and TPSR of methanol on molybdenum oxide without reoxidizing the catalyst, so I chemisorb methanol and see what happens, chemisorb methanol and see what happens. Well, what happens is that it will form formaldehyde. Next time, it will form formaldehyde. Next time, it will form formaldehyde, but I'm running out of oxygen, I have a problem. Why? Because you don't have such an oxygen mobility within the lattice. If you do that on iron molybdate, You put methanol, you make it there, formaldehyde. Another cycle, formaldehyde. Another cycle, formaldehyde. Another cycle, formaldehyde. I can do whatever you want. So why? Because you're also getting the lattice mobility provided by the iron molybdate, so that you get also a more efficient surface. So even when the surface is not like the bulk does have a sedging on it, and this is very important. And these kind of experiments show you the importance of the lattice mobility on these kind of materials. So when you are mixing two materials A and B, is it AB? Yes, sir.
So can you please go back to the slides?
Yes, I can.
The second one.
Right, yeah.
So when you do the reaction, so iron molybdate, so the lattice oxygen is provided to the reaction.
It's compensate, yeah.
But again, that there will be a lag in oxygen, right? For the overall, I mean, the 100 cycles when you do it.
There will be?
So when you do the 100 cycles reactions, so there will be a lack of oxygen, right? Again,
There may be, yeah, but I mean, the oxygen is also feeding the reaction, but this is a more dynamic character. The system is that the system is providing a lattice oxygen.
so how the lattice oxygen is refilled? That's what I'm asking.
I cannot understand you.
So okay, so you have the molybdenum oxide,
Mhm.
you do the reactions.
Mhm.
So during the reaction, the lattice oxygen is utilized,
Mhm.
so that this is not active. I mean, MOO3, molybdenum oxide.
It will not be that efficiently reoxidized. It won't have such a mobility of the lattice oxygen in the system, and the system will be hampered by this oxygen mobility, because in the end the oxygen is going through the lattice oxygen. And if you don't have a good supply of oxygen within the lattice, in the end the reaction will, will be more limited.
Okay, so in the case of the second one, the molybdate, iron molybdate,
Mhm.
so how far that, I mean, the mobility will be there? How many cycles you can do it?
Not sure. But with time, it will be affected. But the point is, with here already with molybdenum, with 3 cycles, you already see things clearly going slow. While in the case of iron molybdate, it can take it quite well. So this is telling you about the mobility of the oxygen within the lattice. This reaction, you are also feeding oxygen, but the migration of the oxygen within the lattice, restoring the sites and everything, will be much more efficient on this iron molybdate than on the molybdenum system. And so this is showing you like only like 3-4 cycles, but you're not going to be running forever with oxygen, without oxygen. You are feeding oxygen as well, but the whole system is much more dynamic. Because you have to be good at oxidizing, which both of them are, but you have to be good also at being regenerated and at getting the oxygen mobility, because you have to recover. And that's basically what this is showing up here. And so A and B, is it AB? Well, depends. Also, the interaction between the oxides is very interesting. When you have 2 oxides, the interface can change. And even if you look at the interface, it's not the same when you are looking at these, for instance, gold nanoparticles on the surface of titania. In oxygen, at low or high pressure, there is already some changes at the interface between these 2 particles. And if you go into reactive environment, you also see that there are changes at the interface. So the oxides are always very dynamic. Yesterday I was drawing that the supported oxides don't look at this like anchoring sites, but more like legs, like spiders running around the surface. The same thing happens with particles, not only with molecularly dispersed oxides. And you saw the movies of these particles moving around. So these kind of things are things that I will show you later on, so I don't have to stop here now. And then something is wrong here for a moment. Moment of panic, don't worry. These things happen sometimes, because this slide shouldn't be here yet.
So would you like to answer? There is a question from online participants.
Yes, because this will help me go back to the wherever I should be. Yeah, I'm listening.
So at a high temperature, is there any change in structure and change of surface morphology? At
For-
high temperature?
For yeah, I mean, I don't know for which system, but typically the answer is yes, as a generic answer. I don't know in particular for what case, but high temperature has an effect. High temperature and reaction conditions has an effect. Even structures may change before. Maybe you need like 600 to get a change in air, but perhaps 300 during reaction.
For both cases, he is asking.
Both cases mean? Because I mean,
Structure and morphology. I think he is referring to structure and morphology.
Okay.
I mean, surface morphology.
In general, what is happening is, if you are heating, things will happen. Like for instance, bulk oxide will spread, or these peroxides maybe aggregate under oxidizing or reducing conditions. But in general, if you are heating, let's go to heating. Well, you are heating and then the oxides will tend to spread, that's it. But no major changes happen. Like for instance, if two phases are there, the possibilities for them to mix will be a given one, whatever, higher or lower. And it requires a higher or lower temperature to get this mixture between the two phases. Now, if you are heating during reaction, things are different, because if you are heating in air, basically things are getting warmer and warmer and warmer, but from the structural point of view, the material is not really changing that much. It's not working, it's just being heated. Now, if you are heating this material during reaction, this material is continuously breaking and forming bonds, absorbing hydrocarbons, releasing oxygen, and that means that bond is formed and broken, and when you form and break, eventually you bond like this, or you bond like that and like that. So the structural dynamic states of a catalyst is much higher when you heat during reaction than without reaction. And now that means that if maybe something happens at 600 in no reaction conditions, it will happen at 300 during reaction, because you are triggering a lot of dynamic states of bonds being formed and broken, and that triggers the reactivity. And an extreme case, mechanochemistry or mechanocatalysis, where basically they just mix powders with high energy, room temperature, and things happen. Oxides get mixed, molecules react even at room temperature, because on these mechanical things you are breaking bonds. When a bond is broken, all of a sudden you have a very reactive site that may trigger a reaction with another oxide or whatever, or with a nearby molecule. And then you are getting reaction because of that. So you need something that destabilizes the system, and then you increase the chances for changes to happen.
I think he's trying to ask what happened to the molybdenum oxide and the iron molybdate during the reaction at high temperature. Was there any change in the structure?
There is, and basically what is happening is that the molybdenum is typically going to the surface, and the surface gets enriched by molybdenum. This is a methanol oxidation catalysis. So typically the surface will get saturated by molybdenum, and then no more molybdenum will migrate from the iron molybdate to the surface. But these kind of reactions, sometimes they volatilize a little bit of the molybdenum. So downstream the reactor, you probably start to get molybdenum oxide crystallites that form because they evaporate from the reactor. So it means that you are continuously losing a little bit of molybdenum, and then the system is pumping new molybdenum to the surface. But usually the surface will have a saturation concentration. And then once that you have the monolayer coverage, you don't get more moly. But because this little by little evaporates, new moly is going back and back into the system. So usually in a methanol oxidation catalyst, upstream, middle stream, downstream, you will see that at the beginning of the bed, they are losing more molybdenum because little by little it's being lost. But the bulk is migrating to the surface. So there is always this kind of dynamic state. Okay, in situ operando in the half hour of the end of the first week. So we are getting into it, and we are getting into it with a Spanish philosopher, Ortega y Gasset. You don't know who Ortega y Gasset was. He died like, I don't know, 70, 60 years ago. But he was a philosopher, a social philosopher. And then he says that the man is himself and his circumstance, which basically is what happens in catalysis, that the catalyst is itself and its circumstance. What he says is that basically you cannot understand the person if you take him away from his social, labor, family, religious, economic, political environment. To understand the person, you have to understand the person in its real working, living conditions. And somehow the same thing happens when we want to understand catalysis. The circumstance is fundamental, because that shapes everything about what a material is. And now, what am I looking at when I'm making in situ studies in catalysis, operando studies in catalysis? Well, the first of all is I get a spectrum, beautiful Raman bands or beautiful crystal bands, beautiful whatever. Is it my working phase? Because maybe you are making a big story of something that is not doing the job. And the other question is, can I trust my activity data because I'm using an in situ cell? Who knows? Well, this is important because if you get data which are diffusion limited or whatever, that is a problem. And then you will be able to say, okay, why is it occurring? How changes in this phase and changes in this activity, how do they connect? Because ultimately we want to get a molecular understanding of the structure-activity relationships. Surface volume is always a key factor. Always remember that, avoid to be fooled. Classical case was VPO catalyst, which is vanadilyl pyrophosphate. Vanadilyl pyrophosphate catalyst is vanadium 4+. But you characterize it and you always see vanadium 5+. That cannot be, it must be wrong, forget it. No, but you have vanadium 5+, no way. Yes, you do. Why? Because the surface of the vanadilyl pyrophosphate is no vanadilyl pyrophosphate. You get dispersed vanadium oxides. We saw it with iron molybdate, we see it with vanadilyl pyrophosphate. With almost anything, you get amorphous layers at the surface, mainly with elements like vanadium, chromium, molybdenum. They like to spread, they like to make a monolayer. And the most important things is that they are at the outermost surface. To be on the surface is fundamental because the molecule interacts with the surface. So you have a reaction with these surface sites, and you correlate with a characterization which is essentially dominated by the bulk. You may not reach good conclusion. You may find, what they say, that concurrence is not correlation. Maybe you may find something and you connect, but that doesn't mean that they are correlated. That may be circumstantial because you are not looking at what really matters. So you have to make sure to see what the molecules are seeing, are interacting with, because this is what really accounts for the performance. That's why it's so important. Good thing in catalysis is that we typically have nanoparticles with trigger to surface to volume ratio. So when you characterize it, even if you get a bulk signal, you see things from the bulk and you can see things from the surface. So you are getting information which is already relevant to what the molecules see, which is what really matters, what really determines the performance of these systems. And I know that you have seen these kind of figures very often, but this is very important to remember that we are dealing with nanomaterials. And if you have a cube, which is 1 by 1 centimeter, total area of this cube is 6 square centimeters, not a big deal. But if you go down into 1 millimeter cubes, the same cube will provide you like 60 square centimeters, which is a nice apartment. But if you take them down to 1 nanometer, then you get like 60 million square centimeters. This is like huge. So you are going from the size of a stamp to the size of a letter to the size of a stadium. And you are still dealing with the same amount mass of material. So the surface is fundamental. What happens at the surface, we have already commented these days. So this is just a quick backup. We have isolated and polymerized species will be like that. You will see the support and you will be the supported phase. Both of them will be available. And above monolayer, you will get nanocrystals. And between the support and the supported phase, you can get some mixed phase, like vanadium and zirconium can make vanadium zirconate, whatever. That phase will typically not be very well defined and is the one that is doing the job. And even when you have crystals or crystallites, remember that probably the surface of it won't be exactly like the bulk of it. All these things are important. Now, one advantage when we are using supported oxides in catalysis is that even when Raman, infrared, many techniques are bulk techniques, in a supported oxide, the supported oxide is all at the surface. So anything you see from that corresponds to the surface because you only have supported oxides at the surface. So even if it goes into the bulk, yeah, you will get a signal from the bulk, but when it comes to the supported moly, supported chromium, supported vanadium, supported niobium, whatever, it will be at the surface. And you will see it because these species are also sensitive to ambient hydration, to reaction, to many things. So this is an advantage. But remember, the technique is typically bulk. It penetrates a few microns into it. Now, the other challenge is to be able to get good activity data, something where you can really connect what I see in the structure with what I measure in the performance, conversion, selectivity, activation energies, whatever. There have been several designs, like for instance these ones over here. They typically in the end they have a catalyst bed, there is a big void volume and different kind of designs so that you see. In my case, the one I start first using is the one that Israel has at Lehigh. It was like that. I already told you the other day that you have a pellet, and a pellet is not the best idea for a catalyst because catalyst is usually a powder. If you have a pellet, you have diffusion control. Not all the particles in the pellet may be working catalytically, but at least this sample will be in equilibrium with the gases that flow through at the temperature that put there, and then when you are making the Raman spectra, 90 degrees, remember the reflection of the watch, it takes a lot of time, but you can get information about how the catalyst looks like, how the catalyst is changing under reactive environment. And then you can run the reaction conditions, so you can make it in situ study during reaction. But that doesn't mean really that you are doing reaction, because when you go out at the outlet of this in situ cell and you ask about the molecules, about the reaction, most of the molecules will say, was there a catalyst? Because most of the molecules did not even interact with the catalyst. So that is something that is telling you the big difference between in situ spectroscopy mimicking reaction conditions or in situ spectroscopy during genuine reaction conditions as you determine simultaneously by measuring activity online, which is basically the idea of operando methodology, because it means working. That's why when we talk about the universe of in situ techniques, you can make some kind of a graphic where on the x-axis you are talking about conditions, whether low temperature or pressure, room temperature or pressure, variable temperature and pressure, working temperature pressure conditions, whatever. You know, like getting increasingly close to the catalytic chemical reactivity. And at the same time, you can also be dealing with studies where there is no reaction, where basically you have some chemisorption, or maybe you do a treatment, or maybe you do a reaction, or you mimic, you provide the conditions, the environment relevant to a reaction, but it's not really a reaction because most of the molecules do not interact with the catalyst because you don't get real catalytic data. Or finally, when you go into genuine reaction conditions, that's how we go from in situ in general to some variable program in situ, reaction in situ where already brings you very, very important variable information, or ultimately when you go to operando conditions, remember not in operando, but operando, where basically you are characterizing things that happen during reaction. Okay, so the key components is basically when you are making operando studies, on the one hand, you are making advanced in situ spectroscopy, reaction in situ, sure, but reaction in situ, not in an in situ cell, but in a catalytic reactor. That ensures you that you are getting catalytic measurement of the performance of this material. At the same time, you also need the contribution of chemical engineering because sometimes you will be dealing with shaped or bigger catalysts, so you have to make sure how you design these things. And of course, you have to characterize the relevant phase. You have to be aware about what is bulk, what is surface, what is relevant, what is irrelevant, but that is part of the research. And all these things is what they blend into operando methodology. Are we there yet? No, because you need the rationale behind what is happening. So when you do these things, in the end, you sometimes end up with some questions. And very often, computational chemistry, DFT, molecular dynamics, whatever, is helping you understand what is happening, model what is happening, and getting the insight. And this is another thing that we will also see and say during operando studies. And ultimately, and you have already seen, is harmonization, standardization, fair data. Do you know what is fair data? Good data, no, because this is capital, these are initials. Fair data means findable, accessible, interoperable, reusable data, because the tendency is that digital data flows of all kinds of experiments, all kinds of measurements will become increasingly standard. Funding agencies in Europe, in the US, in many places are already asking to make the data available. But you cannot give a bunch of data, because a bunch of data is what I do with this. It's like you drown in data, but you are starving of information. So you need that this data comes with the metadata, describing the experiment, the conditions, whether this is a spectrum, whether this is an activity, who made it, the conditions and everything, because that allows you to process thousands or millions of data. Actually, fair data, which is findable, accessible, interoperable, reusable, is also being rephrased like for artificial intelligence ready, because in the end, what they want is this data to be machine readable. They say, okay, tell me what is the best catalyst for this based on all the literature which is published. That is impossible now, because you have to read the papers, you have to copy the data, you have to think, you have to analyze, you have to make a mistake. Oh no, it was like this, whatever. And the idea is the data, as of now, they will be provided in a manner that a machine can go through all the data and process those data. And that will be an important part. And you already saw in Raman why this is important. But okay, going back to the operando concept. So the idea is not that we have an in situ cell where we can make spectra, but a catalytic reactor where we are running a reaction and then we can make the spectra in the catalytic reactor. And therefore we get the real activity data and the structure. And also because these blue molecules are being converted into the green and yellow ones, and here everything is blue, and over here is a progressive change, you will have gradients along the catalyst bed. And then you should also be able to make gradient studies along the catalyst bed. What is happening in it for powder catalyst, for shaped catalyst, and for whatever. And that's why we came with this operando reactor. The first one we made was in 1998, but it is essentially the same because the design we had was very simple, and we haven't found something that is more convenient than that. It's just a UV-Vis cuvette connected to two cylindrical quartz tubes, and then you make a fixed bed. Why a UV-Vis cuvette? Because the windows have optical quality. This is made of quartz, it can take any temperature. And then you have the catalyst. This yellowish powder is a catalyst, the cerium oxide in particular in that photo. And there is the catalyst bed, this is Vanadium ceria. And then you get the black stuff upstream and downstream. What is that black stuff? Well, that black stuff is silicon carbide. Has a very low surface area, very good thermal conductivity, and we want to make sure that the temperature in the catalyst bed is very homogeneous. So it has a heating element around it, but also upstream and downstream this silicon carbide ensures a good temperature on it, and in addition is also assuring a good mixture of the gases before getting into the catalyst bed. Temperature of the catalyst bed is measured because the thermocouple goes into the catalyst bed, and all this thing is heated. Okay, this is just, you see, it's completely transparent. Thermocouple goes into it, and this is the one in 98. We call it the rudder for whatever reason, but essentially the one that we have today is the same. And the nice thing is that you get the gas inlet, the gas outlet, mass spectrometry, infrared, gas chromatography, whatever you want to analyze, and all these things get a heat cover, goes under the Raman microscope, and then you can have Raman observing what is happening in the system. Also, because this is quartz, we can use the same reactor for operando UV-Vis. You cannot use it for infrared because quartz and sapphire is not transparent for infrared, but for infrared there are other configurations, other systems. And we are doing operando because we do catalysis, but we do catalysis because it's a fundamental research, but it is an applied research, and at the end of the day, what you are doing in your laboratory will end up in a larger scale, maybe in a factory, maybe in a micro pilot plant, or in a pilot plant, or whatever, and then you have to measure. So this is also very important to work on shaped catalysts. So we have cells that we are using for shaped catalysts like honeycomb catalyst, and where you can measure it. So this is the size of it, but these things are totally relevant to large-scale catalytic systems. This is for Raman. You can do similar things with infrared, UV-Vis, other technique. For instance, these kind of cells provide a dome that can be made of infrared transparent or UV-Vis transparent, so you can make Raman, you can make UV-Vis, you can make infrared spectroscopy on this, mainly in the configuration of diffuse reflectance. We used this many years ago, and it had a problem that the gas inlet was going in there with the catalyst in there, and something as simple as that resulted in very poor diffusion of the gases on the catalyst bed. So we added this grid, so the gases will expand here and will evenly go through the catalyst bed. Just by doing this little modification of the Harrick diffuse reflectance cell, we managed to get activity and selectivity in the operando cell equivalent to the fixed bed cell. This is a work that I did not for Raman but for infrared, and we were using like organometallic clusters. And then a group of Monier made a quantitative analysis of these kind of studies that we just simply did and it worked. He made a quantitative analysis about the diffusion of gases, about the temperature profiles, and about all the problems, because this kind of system, they had a big void volume and it is a big headache. But in any case, when we did that in our system, which was an organometallic cluster with infrared, and that was back in 1994, Okay, don't tell me how many years ago it was, I know. Time flies. So what we were looking at, it was an organometallic compound, this tricobalt nanocarbonyl compound, which has a lot of carbonyls with cobalt, cobalt, cobalt connected, and at the core you had other element that could be cobalt, could be nickel, could be zinc, could be molybdenum, and this is an organometallic compound. That means that all the metals are saturated. They have carbonyl ligands, they have carboxylate ligands. Now what we did with this cluster is to heat it and to decompose, so that little by little the carboxylate groups go out, the carbonyl groups go out, and then the metal becomes increasingly exposed. At the end of the day, you get a porosity, a metal oxide highly exposed that is very reactive. So when you were heating that material in helium or in hydrogen, basically it was breaking, it was decomposing, so that you are losing all the compound of this structure. And if you were feeding the reaction feed, which was butadiene or crotonaldehyde, we were converting a diolefin into a monoolefin, or crotonaldehyde into crotyl alcohol, hydrogenation reactions. Basically what happened is that at the beginning you got the organometallic cluster with the carboxylic group, the carbonyl ligands, and whatever. And as you increase temperature, you see how the material is decomposing, all the bands of the carbonyl and carboxylic groups are disappearing, and interestingly, the infrared bands of the carbonyl groups start to evolve into something different, which is the infrared band of absorbed carbon monoxide. So the carbonyl ligand on an organometallic compound all of a sudden starts to look like CO chemisorph on a metal. So you see the birth of a catalyst and the death of an organometallic compound. Organometallic compound is no longer there, it's becoming like a metal, and the few ligands that remain, they start behaving like chemisorph CO. And at the same time, because we were feeding propylene, butadiene, you start to observe the absorption of the hydrocarbons, how they are being absorbed at the surface of the material, and you start to observe hydrogenation products. So you see the birth of a catalyst through the decomposition of an organometallic compound. And that was long before the term operando was coined. And this is something important, because the word operando is more recent. We proposed it in 2000. But the idea, the concept, going from I assume the system is working to knowing that the system is working, measuring the online activity, is something that has been done from before. And that is something very important, because when you are making an operando study, you want to make sure that you get conversion that is consistent with the conventional reactor. This is conventional reactor, operando reactor. In this reaction for ethane or propane ODH, at a given temperature, you get a given conversion, and given selectivity to propylene, and given selectivity to CO, CO2, and even the activation energies can be calculated. You cannot calculate apparent activation energies if you have diffusion control. So all these things are telling you that you are doing something which is catalytic, meaningful, and at the same time you are getting the structure. This is the structure that you get from the operando reactor. This is the activity you get from the operando and conventional reactor, totally consistent, and this is the activation energy you get from the operando reactor, which is consistent with the conventional reactor. 2002, we made 3 papers proposing the word operando, and so if you look in literature, there were 3 papers in 2002, the 3 papers I published. Nowadays, we have like 1,000+ papers talking about operando methodology. So it really became something widely accepted by the community. It is important because sometimes the words create realities. The concept was there. People was aware that measuring the activity in a meaningful manner was important. But coming out with the word operando is kind of triggered. Okay, this is true. Because people say, I made in situ studies during reaction as I confirm by the simultaneous activity measurement, and I confirm that this is consistent with my reaction. You lose the attention. People go, what is he saying? Now you say operando, and people know all the ideas behind the term operando. And that's why the idea precedes the term operando. And it is important to have proposed the word operando, sure, but it is more important to acknowledge those who did it from very long ago, before the word operando was coined. And like I told you the other day, Baryashkov in Russia, Novosibirsk, he was doing the first operando study, as I know, back in 1966. I was 1 year old only. So that was long ago. And in this case, he could already rule out the mass Krebs mechanism. This is a reaction for oxidation of SO2 with a vanadate system which is working in molten salts, which is something like Sagar was telling the other day about molten salts. This is a system that is working on molten salts. And you can see perfectly Arrhenius plot. I don't really want to get into the details, but if you are talking about operando studies, you have to go back to those times. In 1995, they made a follow-up 30 years later with a group of Rasmus Furman in Denmark and in collaboration with people from the Baryeskov Institute, because Baryeskov was already there, but the institute in Novosibirsk takes his name, so Baryeskov remains now like the name of that center. And they were talking about all the mechanism of the activation of this system. So that was amazing. But also Charlie Hill, another professor near Chicago, he was looking at hydroformylation on molybdate catalyst, and he designed a cell in the 80s, 1985, which looks like this. Yeah, 1985, exactly. And basically it was a cell where he was running hydroformylation, which is a reaction happening in the liquid phase out of a propylene, and he was looking at two reactions, propylene oxidation and hydroformylation. And he was doing those things in 1991 and 1985. He was also another pioneer in these kind of studies. This is with Raman. The other one was with EPR. Infrared, Lavallee in France, in Normandy, was also doing a lot of in situ studies already reproducing catalytic conditions back in 1989. This is also operando study. This is also very important to keep these things in mind. X-ray diffraction. Haldor Topsøe, the company of Haldor Topsøe, in the times that they were looking at X-ray diffraction, they were making also operando diffraction, operando X-ray diffraction reactors, measuring the online activity, and it was capillary reactor, and they were measuring what was happening on it, and that is back in 1991. Dieter Lemestel and the group of Helmut Nexinger and also the group of Robert Schlugel, they were also doing things like that with Raman, trying to get like double layer systems to observe the layer of the catalyst, because they wanted to look at the fresh catalyst and a catalyst downstream the catalyst bed. So they were using like different kind of configurations to look at different layers, and they were measuring activity online. So that was operando again. So Michael Weaver, electrocatalysis with the platinum, rhodium, they were doing those things. And there is another important aspect, another challenge in catalysis. And this is about the fresh catalyst, age catalyst, and actual working catalyst. They are not the same. A fresh catalyst may look something maybe like this. Okay. An age catalyst after reaction, it is like that. Either way, here or there, you cannot tell the rules of reaction, what is the active side, what is happening, you don't know anything out of that. So you are very limited here, and you have to get into something. Now here, that will be an ideal operando study. You get the active side, you get the molecule, you get what is going to be the mechanism, like kicking the ball, everything. You have everything. Now, can you see these kind of things doing operando spectroscopy? Yes, well, I mean, not exactly this, but you know, the concept of this. But in reality, in most cases, the reaction is something like that. It's much more complex. Now, which one is the active side? This is something that Olivier Marie, a professor from Normandy, presented from the Laboratory of Catalysis and Spectroscopy. And here you have the reaction intermediate, which is the ball, the active side, which is this player. Here you have a poisoning spectre. There you have some spectator species. Now, when you are taking the spectrum, you get everything. You get the spectators, you get the poisoning, you get the active side, you get the molecule. How do you tell apart those things? You look at this and you know what it is, but imagine that you get peaks. And that is the reality, and that is the other challenge. Okay, I'm taking a spectra. What the hell I see here? I get activity. Okay, I know the activity. Now explain this activity with this spectra. Now that is the big challenge. But once that you can tell apart what is the active side, the reaction intermediate, the poisoning species, which are the spectators, because the spectators will give you a lot of bands. How do you tell those things apart? Once that you know those things, then you can investigate the aging. You can also develop techniques to avoid the activation phenomena. There are many things that you can do, but you need the knowledge, you need the insight. And these two slides from Olivier Marie in France are fantastic. So I'm just taking the liberty of using these two slides because I really I was trying to get it from cricket, from here, which is more. You play cricket is the most typical here, but I guess you are acquainted with football at least to understand about this kind of and rugby and so on. Okay, time. Okay, so we are already ready to digest this weekend and enjoy the weekend thinking about operando things, or having tomorrow laboratory classes with some hands-on session and so on. Okay. Okay, well, okay, fine, I got it. So that the spectra looks similar, but the peak width is changing from one position to the other. Okay, in that case, basically this is telling you that in that part where you are looking at, the system is more defective, it's less defined, or it has like a smaller crystals, because you have like a broader peaks. So you can tell the homogeneity of the sample. So basically, but in that case, it won't be necessarily, well, it could be confocal, you can look inside, or can be just at the surface. But basically, at given positions, you will see changes, like you will always see the same phase. Let's say iron molybdate. But sometimes the peaks will be sharper, and sometimes the peaks will be broader. So basically, this is telling you that there's more defects, or there are more vacancies, or there are more defects at different positions of this material. Probably topological effect would not be. You must also bear in mind that you're looking at a sample, you're looking at a few microns, and the sample will be millimeters or whatever. So basically, it's telling you that the sample is not absolutely homogeneous. Basically, when you have a sample, of course, because heterogeneous catalyst, or typically, I mean, when you make an operando study, you finally look at the specific area. But before doing operando things, what we typically do is to make maps at 100 points or whatever, just to be sure that you get something representative, that you're not looking at all of the table and then you focus on this, because this spot is not the table, and then you make all your study here, and you compare this peak with this peak. Oh, it looks very different. Yeah, but if you look at this and this and you make 100 points, you will see like 99 are like this and 1 is like that. So this is an impurity, or this is another phase, or this is whatever. So you always need, because when you make this kind of assessment, it has to have a statistical minimum. With a couple of points, you can be looking at very odd spots. In all the table, you look here, and none of them is the table. Or you look here and here. So maybe here and here you see little differences, but you're dealing with 2. So it's better to have 10, 20, or 100. And now this is more easy, because with the mapping capacities, you can look at many positions before you make this assessment to be sure that it is statistically relevant. And then you will see always a variability. You will see materials better defined or worse defined, but there can be some variability associated to different kind of reactivities. Like we will be talking about Syria, and there are some interesting things on Syria that could be relevant to that. And so you will see that on Monday, I guess. But yeah, I mean, couple of spots is a couple of spots. It's like I say, I met 2 people from India, and they are very funny, or they are very angry, or they're very serious. 2. You know how many Indians are there? So it's not a representative sample. You need more samples to make a good assessment. From an unknown sample, it's difficult to tell because you never know. There are many problems. Some of them may give you fluorescence sometimes, and that is, if I look for that, I can look for that. We made a colleague. I made a spectra for a colleague in my institute. It was a catalyst which was a zinc oxide on graphene, and then we were making a map. And we were making a map with a green laser and with a violet laser. And it was very interesting because with the green laser, we could very nicely see the graphene Raman bands, but the zinc oxide Raman bands were very weak. And then we make a map with the violet laser, and we could see very nicely the zinc bands and not that well the graphene oxide bands. So we have like 2 maps, the green map and the violet map. And on each one was used to discuss about one or the other, and we were looking at the same area. So you keep the same area of the sample, we make this, and then we change the laser, and on the same location, on the same coordinates, we make the other map with the other laser. So a priori, sometimes you don't really know. Typically, it will be okay. Now, when you are dealing with mixed phases, sometimes you may get this kind of phenomena that one looks stronger than the other, or vice versa. But that's very phenomenological. That will depend on experience. I like to go for the green and violet laser because of the kind of samples I'm looking at that's convenient. People who look at organic samples, they like to go to the near infrared, but it typically depends on the availability, because usually a typical Raman system will have 1, 2, 3 lasers. If you have one laser, that's it, no choice. If you have 2 or 3 lasers, you can play and find out which one is the best. But it's difficult to tell a priori, without any experience, which one is best. I mean, you will see in papers that people is using this or that one. And also because of this lack, this problem of harmonization, also because of resonance problems, sometimes you find like the literature of a given system is focused on a given laser. Like people looking at carbon nanotubes, very many papers use near infrared. And if you look at the carbon nanotubes with other different lasers, some bands change. For instance, in carbon nanotubes, you have like the tube. So this nanotube will have a lot of vibration, and one of them is radial breathing mode. It's like your chest. So it's like it goes bigger and smaller on the vibration. It's like breathing. So this radial breathing mode. With near infrared, it's very well described, very well commented, because it won't be the same for a single layer monotube than that for multi-layer monotube, where you have multiple layers, because that will be affected. These kind of things, for instance, are very well described with near infrared, but you are using green or violet and you hardly find any literature. So, and sometimes you look at it with another laser and some of the peaks don't look exactly the same, which is part of the problem of harmonization. That's why we are starting to look at these kind of processes, because sometimes it is the response, sometimes can be some moderate resonance, sometimes can be many artifacts, so it's tricky. But which one is the best Raman? You may look about what is the most frequent laser for this kind of sample, but that doesn't really mean anything, but that can be a good approach, but you don't really know. But I'll try to remember and look for the spectra of this zinc and graphene, because that is an interesting example. Thank you.
There is a question from
Sure.
Can we do the in situ reaction study using, without any vacuum inside the institution?
Without
Any vacuum?
vacuum. Most of the reactions that you do in catalysis are usually not in vacuum. That depends on the system. I mean, if you are talking with Raman, UVVs, infrared, we are doing atmospheric pressure, high pressure, and that is not a problem. If you want to make operando study of XPS, or if you want to do operando study of transmission electron microscopy, you need vacuum, because the technique works in vacuum, but in your sample, you need pressure, because you're doing the reaction. So I believe the question is because of that. So if you're looking at the catalyst, like we saw yesterday, these transmission electron microscopy reactors, well, this reactor is very, very small. It's smaller than my finger. So you get like a metallic system with a window that could be graphene window or whatever, something like that. So you get a metallic piece here. Over here you have your sample, the flow going through, but this is very small. This is like a few millimeters, and over here you will have liquid, or you will have gases, but then you have all the electron beam for the microscopy here, and everything here is vacuum. So if the whole chamber is very big and everything is vacuum, and you only get a little bit here, and you are dealing with a few microns here, and the whole system is only a few millimeters, but the reactive area is, and then this is good enough for the electrons to go through and reach the detector. For XPS, it will be the same thing. You will have the sample with your reactor and whatever, X-ray will come out, and then what happens is over there they have like metallic windows. So here they will have a little bit of pressure, and over here they will have high vacuum. Basically, the problem is that because of these little holes, the pressure will very difficultly diffuse there and will be vacuum. Because this is important, because the electron that come out and go to the XPS X-ray photoelectron detector, they must run in vacuum, because if there were gases here, these electrons will never get to the detector and you will never get the information. So they keep a very nearby area where there are molecules and where you have catalytic relevant environment. But this is the big challenge. XPS, near atmospheric pressure, like this, transmission microscopy, like that. But then you go to NMR, EPR, Raman, X-ray diffraction, all these things, you can do it not only at atmospheric pressure, but at high pressure, or even supercritical conditions.
So in the sample preparation, you recommended not to make the
Sorry?
the sample preparation.
Yeah.
So you recommended not to make the pellets due to the diffusion limit. So it,
Well, but diffusion control is basically, I have one slide for next week, but the diffusion control will be the same. Is for a given residence time, you are increasing the weight and the flow of the catalyst at the same time to keep the same residence time. And if everything is reproducible, you don't have external diffusion control. And for the particle size, you keep all the conditions that you're decreasing the particle size, and as long as everything is the same, that's okay. If things start to decrease, then you have internal diffusion controls. But that will be, basically the same principles that apply to normal catalysis and engineering apply to the operando reactors. Because basically you are doing spectroscopy in a catalytic reactor.
so if the sample has a less intensity, if it flies during the analysis, what we can do?
Then probably this sample is also flying during your catalysis reaction in the conventional reactor. Because you have the same system. Samples flying under control is a fluidized bed. They are just flying in a fluid bed. But there are operando reactors that are looking at fluid bed. In such a case, you have the fluid bed. So you get the flow, and then the particles are not loose, but just moving around all the time. And then you are making a spectroscopy from whatever technique that you want to make. You are looking at the spectroscopy there. At which position? Well, it is a fluid bed, so basically it will give you an average value because it's continuously moving, but that is okay, it gives you an average. Some people is using even the fluid bed, even if there is no need for that, with UV Raman, because with UV Raman the photons have a very, very high energy. So photons really can burn your sample. And by doing that, you minimize the exposure of a given particle to the laser, but many particles are passing by, so you get the average, and the sample never gets damaged, because when you look at the wafer with the UV laser, the sample is like this before the analysis, and then after the analysis, the spectrum was made there. You see like a black spot that it was burned. You avoid that. But for many reactions, you get fluid bed. But now reactions where your sample flies away, industrially, you don't have those because you run out of the catalyst right away. So usually, in any case, the catalyst in a reactor, even if they are a powder, they will be in a bed, but there will always be like a porous plate, upstream and downstream, so that in the end the gases go through this porous plate, and here you will have the catalyst. And of course, if it's very small, they will plug it or they will flow away, but you never do a reaction with very fine powder, because when you go an industrial reactor with very fine powder, it will fly away. And if it doesn't fly away because you avoid it flying, it will plug, and there will be hardly no porosity, and then in the end the gases will only go through preferential channels that at the end go, but then most of the catalyst will not even interact with the gas. So usually you need a particle size that is not too small to avoid internal diffusion control, to avoid a problem of flying away, and to avoid many things. So nobody will have it very light that flies away. And it will not have it very big because there is another kind of diffusion.